Hello everyone. Today I will talk about CD8 positive cytotoxic T cell. Now in one word, CD8 positive cytotoxic T cell are reserve army branch of the immune system. So they are ready for combat for viral infections. Let's just take a look at from where the CD8 positive T cells develop. So just like normal T cell or B cell, the development starts from the bone marrow. And everything starts from hematopoietic pluripotent stem cell or HSCs. From hematopoietic pluripotent stem cell, they develop into lymphoid progenitor cells. The lymphoid progenitor cells give rise to precursor T cells. Now precursor T cell then enter the bloodstream and migrate towards the thymus gland. Inside the thymus, there are sequential stages of development of a T cell. In a particular stage, known as double positive stage, the T cells, the immature T cells, expresses both CD8 and CD4 co-receptors. Now, depending upon whether they are recognizing MHC1 bound peptide or MHC2 bound peptide, they differentiate into either helper T cell or into cytotoxic T cell. Now, if they can recognize MHC1 bound peptides, then they would downregulate CD4 expression and upregulate CD8 expression. Ultimately, it would switch its fate towards cytotoxic T cell. Whereas, if it recognizes peptides bound in MHC class 2 molecules, it would upregulate its CD4 expression and downregulate CD8 expression. Ultimately, it would become a helper T cell. Now, once the cytotoxic T cell is formed, it is still naive. It doesn't recognize antigen yet. In order to be mature, it need to learn how to recognize antigen from dendritic cells. And also, another trigger of interleukin 2 helps these cytotoxic T cell to proliferate and expand clonally and give rise to a lot of cytotoxic T cell. Now, cytotoxic T cell provides immunity against viruses. So let's just take a look how cytotoxic T cell fights against the viruses. Now, here are some epithelial cells which are infected by these viruses. Once the virus entered this cell, the virus injected its genome, and the genome is successfully replicated, transcribed, and translated. And ultimately, viral proteins are formed. Now, cells, inside the cells, there are always degradation machinery for proteins, known as proteasomes. Some of the viral proteins are also degraded by these proteasomes. Here are some viral proteins which are degraded by the proteasome. They enter the endoplasmic reticulum via TAP transporter and loaded onto class 1 MHC molecule. Eventually, the class 1 MHC molecule would be displayed on the surface of the cell containing the viral antigen. Now, CD8 positive T cells are always looking for viral peptides displayed on class 1 MHC molecules. Now, CD8 positive T cells would secrete granzymes and perforin and ultimately lead to the apoptosis of the virus infected cell. And that's how it would save the nearby cells from further infection. Let's just take a look at how a CD8 positive T cell kills its target. So CD8 positive T cells or cytotoxic T cells use two different strategies to kill its target cell. One, it secretes perforin and granzymes which get endocytosed by the target cell, and perforin leak out from the endocytosed vesicle, and perforin can interact with mitochondrial pro-apoptotic proteins known as bead or BACs. Now, these perforins and bead make the mitochondrial membrane porous, such that cytochrome C could be released, and cytochrome C is not expected in the cytosol. Once present in the cytosol, cytochrome C can interact with APAF1. Now APAF1 is one of the bad players. APAF1 cleaves inactive format of caspase 3 and makes it active. So here is an inactive procaspase 3, which is cleaved with the help of APAF1 and cytochrome C complex to cleaved and active caspase 3. Now caspase 3 is an executor caspase. Caspase 3 can do a lot of things. They have protease activity. They can further activate several other proteases. They cleave the microtubule. They chop the DNA, and ultimately they lead to programmed cell death or apoptosis. There is another way by which cytotoxic T cell can induce apoptosis of its target cell. Now cytotoxic T cell expresses FAS ligand, whereas FAS receptors are actually expressed on the normal cells. Now FAS ligand interaction ultimately leads to activation of procaspase 8 to an active form of cleaved caspase 8. Now cleaved or active caspase 8 can also lead to further cleavage of inactive caspase 3 to active caspase 3, which would ultimately lead to apoptosis. By these two parallel mechanisms, a cytotoxic T cell can kill its target, and thereby cytotoxic T cell can save other cell from further damage or further infection. And that concludes my video on this topic. If you like my video, give it a quick thumbs up. Don't forget to like, share and subscribe. Thank you.